Well, thank you for the invitation, and I'm glad to be here to talk about our work on axonal degeneration, which you know that for almost all the symptoms that occur from neuropathy are going to be from axonal degeneration, either directly or indirectly. So we focused on that particular aspect. Oops, hold on here. Let's see here. Yeah, I see that now. Okay, disclosures. I have to give you that slide so we don't have any legal troubles. And here are the contributors. So here are the people that do the work in the lab over the last many years. So they really get all the credit. Besides Aaron. So Aaron and I we've talked a lot about cooperation and partnerships. And Aaron and I have had a long-standing partnership, Aaron D'Antonio, for about 15 years now. Our labs are fairly joint at this point, and we do almost all of our work together, including the grant writing and everything else. So, okay, so axonal degeneration, primary driver. So this is a self-destructive process, active self-destructive process. And that was really first discovered with the WLD mice of Hugh Perry in the late 80s. And then Michael Coleman did positional cloning and found out that NMNAT1 was the mutation that really caused this mouse to have this strange phenotype, which was when you transected the axons, they did not degenerate. So there was no Wallerian degeneration in those mice. So that seemed like a phenomenon worth exploring, and many of us did. And so you see here's an example what axons look like in a dish if they're wild type. And you can see what they look like 24 hours after transection. And then if you have this NMNAT overexpressed in them, they magically do not degenerate. So they're severed, but they sit there and they're metabolically active, et cetera. So what is this enzyme? It's an enzyme that converts the mononucleotide NMN, adds ATP, and you get the well-known NAD dinucleotide. So it was a little strange on how would an NAD enzyme be so important in axonal degeneration. And Michael Coleman showed that there was another form. There are three forms of NMNAT. NMNAT2 is largely in the axon and is a short-lived protein, and we've found that it's degraded actively in a number of different circumstances, but a very short-lived protein. So that if any kind of unhealthiness, transport, axotomy, et cetera, happens, you have no NMNAT2 in your axon, and that will trigger some events that we'll talk about shortly. So we spent a lot of years trying to figure out what NAD and neurometabolism might be doing to an axon, and published a few interesting papers, as did others. But at the end of the day, in the genetics department, if you can't figure out what to do next, you do a screen. And so we did a screen. And this is the detritus of that screen here. This was about 100,000 different lentivirus clones that were put into this robot. Josiah Gertz was a fantastic student who was the robot to start with, and then he wanted me to buy these fancy robots. And, you know, out of all that, we got one hit. And Mark Freeman did this similar screen in Drosophila, and he also got one hit. And luckily for the field, it was the same hit. So it's an enzyme called SARM, and here's the initial data that we had showing that SARM was very critical for axonal degeneration. When you have a SARM wild type with SARM, you get degradation after transection, and in the SARM knockout, you saw nothing. And a friend of mine, I'm not being heard well. Okay, can you hear me now? I thought I could project pretty well. A friend of mine at WashU had SARM knockout mice. He was an immunologist. And I went over to talk to him about it. He goes, oh, yeah, those mice are worthless. They don't do anything. We've studied them forever. They don't have an immune phenotype. And I got rid of them. Luckily, his people in his lab pay as much attention to him as mine pay to me. And they hadn't got rid of those mice. So we were able to get them, and we were able to do these studies and quickly show that there was a problem and that you can save axons very nicely with no SARM. So what is SARM? So SARM is a toll-like receptor adapter, and it was came to fame because of its tear domains, which come together and then form a signaling complex when the toll-like receptor wants to signal. So that's what it was for. So it was hard to put that in context then with NAD. It's tethered, unlike many of these receptors, it's tethered to the mitochondrial membrane. And it's mostly just expressed in neurons, which is why immunologists could care less. And this tear domain is highly conserved all the way back to archaea. I won't have time to talk about that, but there's a lot of interesting work going on in tear domains in bacteria as well. So we spent some time trying to figure out the connection between NAD and SARM. And we knew that when you brought these tear domains together, that NAD would suddenly plummet in a cell. You could do that artificially. But exactly what the mechanism was difficult. So we spent several years trying to find this fabled NADase that must be interacting with SARM, these tear domains, which are known to be molecular glue, and we failed. Then Co came to the lab, and he was a fantastic student. He came to the lab, and we had this project basically to say, maybe somehow it's SARM itself. So after only a couple months, really a couple months, he developed this assay where he could just very gently lyse the cell and bring SARM down with beads in a tube. And then he would add NAD. And I'll never forget when he came in my office. It's like one of the most compelling moments of my professional career. He walks in and he shows me this graph, which is why I have this simple thing up here. The NAD, you spike in a lot of NAD, and 5 minutes after you add the enzyme, there's no NAD. And so you worry that it's the pure enzyme and all, and we did a lot of work to prove that that was the case. And so eventually what we got out of that experiment was two things. One, we knew that SARM somehow mysteriously was the NADase. And two, we found that it converted things to ADPR and cyclic ADPR, which ends up being a biomarker for SARM activity. So it was a fairly good 2 months for Co. So this NAD hydrolase activity, as it's called, is conserved. So it's conserved in plants and is important in immunity in plants. It's conserved in bacteria and is important in phage immunity in bacteria. And many alternative products besides the ones I showed you have now been discovered. But once we knew it was an enzyme, enzymes are tractable, right? They're inhibitable. And so we thought that we were on the right road then to be able to find something that would work. So we formed a company, Disarm Therapeutics, co-founded that, and they went on to find a number of small molecule inhibitors, of which I show you on this slide how effective they are in culture. So this is when you do transection plus the drug, the SARM inhibitor. You see good axons after transection, or you can do it with rotin and mitochondrial toxins and see again that they preserve the axonal integrity if you block the SARM. So we knew that SARM was an NAD hydrolase. We also knew that most of you sitting in this room right now, your SARM must be off in your axons because you're in pretty good shape. For others with neuropathy, SARM is probably on low levels, and we want to talk about that in a moment. So we knew that it was auto-inhibited. We knew that if we cleaved off the amino terminus of the enzyme, it would be constitutively active. So we knew there was some regulatory component there in the amino terminus, which is important also for therapeutics. But we were stymied in exactly where it was and how it worked. Then we teamed up with Boston Kobe and Thomas Vey, and they did cryo-EM with us. And this is the cryo-EM of the SARM octamer. You see here the SAM domains that bind it all together are in the middle of the core, and then the tear domains that are the enzymatic activity are outside here, separated normally by the amino terminal domain, but then when they become active, they swivel and they become dimerized, and then they become active enzymes. So that's how it works. And here's in cartoon fashion what the discovery that really united the field, if you will. There were There were camps that said that SARM was activated by NMN, a precursor of NAD, and there were others, this was us, that thought that NAD itself fed back on SARM and blocked its activity. And it turned out, as was really nice for the field, that both were right. So NAD normally fits into this pocket in the immunoterminus and blocks SARM activity. But when NAD levels fall and NMN levels, its precursors, rise, then NMN predominates and binds into that pocket, that high affinity, and that changes the conformation and SARM becomes active. Now, I want to just put all these, the work that we've done and others have done into context and try to order the events in this axon defragmentation. And this is injury evoked. So this is single axon. We're looking at single axons throughout this. So 50 to 100 single axons were time-lapse photography, and we did lots of work on this. And Klein, we did a great job on this. The bottom line is that SARM gets activated. A few minutes later, NAD is depleted. Then shortly after that, ATP is lost. Then mitochondria become less mobile, less active. Calcium becomes the influx, and that's a critical moment. Once the calcium starts to come in, you get flipping of the membrane. So phosphatidylserine flipping, and then a few 60 minutes after that, you start to see those nanopores where dextran comes in, and you know the axon is beginning to fragment. So this is sort of the wave of molecular changes that occur in this axon fragmentation. But this is acute. This is injury drive. What happens in disease? What happens in chronic SARM activation? So I want to tell you a little bit about a couple of discoveries we have that deal with this idea of what might be going on in chronic disease. So these were two patients that were brought to us by Sabrina Young at CHOP, and they had mutations. They had a disease that was like chronic inflammatory polyneuropathy. Their initial presentation looked almost like childhood Guillain-Barré, which is, less than 1 in a million patients. So it seemed like that would be relatively unlikely. So I give you some of the details of their clinical course, but the bottom line is that it was a disease that was much more rare than 1 in a million. And it was in mutations in NMNAT2. So there were 2 mutations. There have been a couple other patients described that were stillborn, and these patients survived. They have about 2 alleles, 2 different alleles. One has 12% activity, one has 2% activity. So they're dealing with 5-10% activity. They've been in wheelchairs their entire life, but interestingly, they have no muscle mass from that. They're 25 years old now, and they're in college. So the CNS and the PNS are behaving very differently, of course, in response to these mutations. So we set out to model this. So we made mice that mimic those mutations, and then we studied those mice. And I'll just briefly go over what we found. What we found is that they quickly decline in terms of their motor strength, but never have any sensory deficits. You see here on the right-hand side, the changes in the femoral are quite dramatic, but the changes in the sural are much less obvious, as in keeping with their lack of sensory deficits. It wouldn't be a talk from me if it didn't talk about SARM a little bit. So we mated these mice to a SARM knockout, and lo and behold, they became perfectly fine. So the marker for SARM, cyclic ADPR, goes down. Of course, you don't have any SARM. Their distal muscle mass recovers completely in the absence of SARM, as does their strength. So this is a SARM-driven disease. And what we noticed, though, when we were investigating this, and we had a person in the lab who was also doing single-cell sequencing of nerve macrophages and microglia, and he just was staining anything he could find. And so he stained these nerves of these animals with CD68 and found this very striking increase in macrophages in these nerves. especially angry CD68 positive ones. So I know I told you that the sensory and the motor were different. When we looked at the sural nerve, we didn't see any of these macrophages. In the femoral, which is mostly motor in mice, well, we did. But curiously, the marker for SARM activation, cyclic ADPR, was the same in both nerves. So there's a complete disconnect between what we see in terms of neuroinflammation and cyclic ADPR. We have not figured that part out yet, but what we have done is some depletion studies. So we wanted to know whether these macrophages were helping, were beneficial, or were they detrimental to the disease. So we used a CSF receptor antibody, as is common these days, and looked at what would happen. And when you deplete the macrophages early on, these animals get sick about 2 months of age. If you deplete it early on, we could rescue most of the pathology, both the strength and the nerve degeneration, axonal degeneration. We also did this later on because there's been talk about 2 minutes. Okay, there's been talk about SARM and it's when you preserve an axon by inhibiting SARM. There's been talk that, and I think people know about this show, The Walking Dead. So there are people that want to call these zombie axons, so that they're preserved, but they're not functional. Here, when we deplete this later on, we see that these axons can recover. So they're stressed, but they're viable. And I think it's important for these therapies. Now, the last 2 minutes, I want to talk about CMT2A. So there's a rat model of CMT2A that was generated by CMT Association. And we got that model and we made SARM knockout rats. And we pursued those animals to see whether SARM knockout, whether inhibiting SARM would be useful in CMT2A. And you see here the pathology in the tibial nerve. Oops. And you can see quite clearly that there is really a lot of pathology in that nerve in the animal, but in the SARM knockout, much of that is recovered. This is at 1 year of age in these animals. Their electrophysiology also recovered. I'm not going to show you that, but I am going to show you the neuromuscular junction. I think it's quite dramatic. You see the nice fimbria here in the wild type. This is all basically occluded in the animal. And then in the SARM knockout, much to our surprise, much of the neuromuscular junction is now, again, intact. And finally, in a puzzle that we still don't understand, we noted that early on that mitochondrial inhibitors, so mitochondrial dysfunction, would activate SARM1. We don't know the mechanism yet, but we know that it occurs. And so we were extremely gratified and surprised by the finding that in the MFN2 rats, which have of course, a mutation in a mitochondrial protein, that is the direct problem, is lack of fusion and fission in the mitochondria. And yet, when you knock out SARM in those animals, those mitochondria recover. They recover quite dramatically. So we're trying to figure out how that occurs, but we think that this SARM mitochondrial feedback loop is an important one for many neurodegenerative diseases, including CMT. So with that, I was supposed to give you a few future directions, but here they are, and we can talk about them later. Okay. Thanks for your time. We do this handoff.
Thank you so much. We have one question. Is it okay? We have maybe one question, and then if you have more questions, fine.
Yeah. Bruce.
Thanks, Jeff. I was wondering if you think that SARM is playing a role in all forms of axon degeneration, or are there areas or types of degeneration when it doesn't? So, for example, on that Hope crossed the SARM knockouts with CMT1A, and it did not rescue.
Right. So the answer I had on one of my questions, what are the parallel pathways? SARM independent pathways. So there are going to be some. There's been numerous examples of neurodegenerative models mated to SARM knockouts that don't improve. So I think there are going to be other pathways, and we're actively looking for those now that we've got one kind of partitioned off. So yeah, the answer is yes. I'm sure there are.
All right, so just to, stay on time, we're gonna go ahead and go to Dr. Rick Dobrowsky for his presentation.
Okay, so Dr. Milbrandt's a big, tough act to follow, but I will do my best. Okay, and we're going to kind of shift directions from fundamental biology of neurodegeneration. I'm going to tell you a pharmacologic story about disease modification approach to treat CMT1X by a mechanism we think involves modifying a heat shock response. And so I need to thank the people who actually do the work. I'm glad to hear that Dr. Milbrandt's people don't listen to him as much as my people don't listen to me. So that must be a common thing among graduate students and postdocs. I need to thank Suki Kerr and Allison Zhang, who did a lot of the work I'm going to talk about. And then Rudolph Martini and Charles for providing some of the mouse models that we used. This work was funded by Ignite, grant from NIH, and we partner with Reata Pharmaceuticals. They provided the human formulation of the drug Domesviv that we developed at the University of Kansas and licensed to them. And I need to do the conflict statement saying, given the statement provided me by Reata, which essentially means I have a financial interest in this. So I'm going to talk about the heat shock response briefly. I'm not going to talk about CMT. You know more about that than me. I'm going to talk to you about our drugs, which are called Novalogs, and this hypothesis we've been pursuing that Novalogs activate the heat shock response to increase HSP70, and this is a key node in producing neuroprotection. Show you some data from 3 models of CMT1X, conclusions, and a little bit of our clinical perspectives. And I see one of our pictures didn't show up, but basically the heat shock response is stimulated by some type of proteotoxic stress in the cell, which leads to the dissociation of a molecular chaperone called HSP90 with heat shock factor 1, which is a transcription factor, which after it's released, trimerizes, go to the nucleus, binds to these heat shock elements, to promote an increase in molecular chaperones and other proteins that help overcome proteotoxic stress. These, the role of HSP70 is important in helping to improve various aspects of different neuropathies. And in terms of the CMT field, Dr. Notopek has shown that if we use these heat shock inhibitors to activate HSP70, they can improve CMT2A. We have used our drug to actually show that it can improve diabetic neuropathies. And so the question came down, since diabetic neuropathies are not dependent per se on proteotoxic stress and protein aggregates such as CMT1A, would they work in another neuropathic disease such as TMT1X, which develops a neuropathy, but not due to any kind of protein aggregate formation. So N-terminal inhibitors of HSP90 have long history. They're promising, but problematic. They block the chaperone activity, the ATPase activity, which is necessary for protein folding, which is one of the key molecular features of HSP90. And they've been explored as chemotherapeutics because if you block the folding of oncoproteins, you can block the disease. Unfortunately, when you inhibit the N-terminal of HSP90, you also release HSF1, which activates the heat shock response, produces more molecular chaperones to antagonize the phenotype that you're trying to promote, which is cytotoxicity of the cancer cell. And so Dr. Notopek, as I said, has shown that we have these N-terminal inhibitors can also provide neuroprotection by activating the heat shock response, but you have the inverse caveat. They can also inhibit protein folding, which can be cytotoxic to the neuron. And so I had the fortune of living next door in my lab to a talented medicinal chemist who was focused on trying to develop new ways to inhibit and modulate HSP90 by targeting the C-terminal region of the molecule rather than the N-terminal region. And he came up, started with an antibiotic called novabiocin, which was the first molecule known to kind of bind the C-terminal, and did some modifications and came up with a couple of generations of what we call novalogs. And the neuroprotective form is called KU32, as you see on your, I guess it's on your right side, the left side of the screen. And KU174, which is a cytotoxic or chemotherapeutic. And you can see the difference in the structure of those molecules relates to kind of the side chain that hangs off the coumarin ring of those molecules. And so by modulating that side chain, we can essentially modulate whether the molecule is neuroprotective or neurotoxic. And we've done a lot of structure activity relationships to kind of discern this using KU32. And you can see here that when the amide chain coming off the coumarin ring is modified just by increasing its hydrophobicity, we could essentially change this molecule from being cytoprotective and not inducing cytotoxicity to becoming more cytotoxic. And so you can see here with the increase in HSF1, as you increase the hydrophobicity, you decrease the expression of HSS1, and you increase the IC50 for cytotoxicity. And so we kind of know that, you know, this amide side chain on the molecule is important, and that we don't really want to, make changes on that to maintain neuroprotection. So we tried to promote KU32 to NCATs to explore its role clinically, and they didn't like the electrophilicity of the coumarin ring. And so the second generation of molecules went and focused on altering that while still maintaining that amide side chain and the noviose sugar, which is important for binding to HSP90. And this came up with a generation called semdamosfib, or KU596. So you can see we went from KU32 to KU596. So quite a lot of molecules were produced before we came up with one. And this essentially mimics the binding of KU32 to the HSP90 protein. And Reata Pharmaceuticals took some interest in our work using KU32, and they licensed this portfolio back in 2013 and used KU596 as the lead compound to go into phase 1 trials for potentially looking at diabetic neuropathy. And it had a very good a profile and no adverse events and was well tolerated. So in the last few minutes, I'm going to talk to you about using KU596 to treat CMT1X. So we see we've used several mouse models here on the left. I'm not going to talk to you about the models we use for mechanism. I'm just going to show you some of the physiologic data for improving or being disease modifying, as our patient panel said they'd like to see. And so we wait to about 4 months of age in these mice when they start developing some motor phenotypes, begin dosing them daily, look at their motor function over time. And at the end of the study, we do some electrophysiology and look at their tissues. And this was recently published in a paper, and so most of that I'll be showing you. And so here you see the results using the 3 different animal models, looking at the grip strength. And we did some dose response studies. And you can see the grip strength with daily treatment of semdamosfib was significantly improved or slowed. You know, it's not perfect, and it's definitely lower, still lower than wild type, which I don't show you on here, but it is slowing the progression of the loss of grip strength in both the CX32-DEF knockout. So the drug is not working by improving CX32 function because there's no CX32. In the T55I mice, this is a gain of function mutant, which causes protein aggregation of the mutant CX32 in the ER. Since HSP70 induction can remove protein aggregates, we were worried that it might not be as effective. But we see here that in the T55I mice, it still seemed to work just as well as in the knockouts. In the R75W mice, it's a little different. That protein accumulates in the Golgi instead of the ER. But once again, the grip strength was still improved. And on the bottom, you can see just the fold control of the improvement versus the 3 doses. I just show you the time course for 1 dose at the top. When we look at nerve conduction velocity, we can also see that it was improved in all the mouse models with some modest differences between the mouse models. It's clearly the CX32 def strain responded the best, but the T55Is responded fairly well as well. When we looked at compound muscle action potential, the results were a little muddier. CX32 def mice did show an improvement in compound muscle action potential, but neither of the other 2 strains did. And so when we kind of just plot grip strength versus nerve conduction velocity, we can see that they kind of correlate very well with the drug-induced improvement in grip strength and nerve conduction velocity. Since biomarkers are a great measure potentially of efficacy, we looked at neurofilament light chain, which is known to be an emerging biomarker in different types of peripheral neuropathies. I think the Cleopas group had reported that it increased in CMT1X, which we did find. However, the drug unfortunately did not decrease it. This was quite crushing data when the student showed me the results. But you can see we used numerous animals and it is what it is. So I'm not sure what to say about that. We did a drug withdrawal study to look at pharmacodynamic efficacy, where we treated the mice beginning at 12 weeks of age with daily drug treatment. We can see that grip strength markedly improved. At that point, the drug was withdrawn and they just received vehicle. You can see a gradual decline, suggesting that there is a prolonged pharmacodynamic effect, suggesting probably some transcriptional mechanisms or a prolonged decrease in neuroinflammation that we think is allowing that drug to work a few weeks after withdrawal. And so this has potential impact on dosing and frequency of taking the drug for potentially seeing functional efficacy. And once again, you can see that with the correlation, the open blue circle is with the drug after 12 weeks. It improved the velocity. The open red circle was untreated drug at that 12 weeks. And then the closed circles are after drug withdrawal, where we kind of lose nerve conduction and grip strength. Obviously, you want to know about myelination. We looked at the femoral nerve, which, as Dr. Milbrandt told us, is a nice motor nerve. There we have a wild type on the left, the CX32 def showing the typical degeneration, myelin thinning in the nerve. And then after 5 months of sindomosvid therapy, improving myelination, we looked at the G ratio, which is a measure of the myelin thickness. And on the left, you can see that the CX32 def that were treated with sindomosvid tend to show a lower G ratio, suggesting an improvement in myelination. We looked at innervation of the neuromuscular junctions. The data here was encouraging, but kind of muddy. Some mice showed nice responses, some mice did not. In the middle panel, we can see a totally denervated neuromuscular junction in the CX32 def mouse. And in the bottom panel, we can see some of the reinnervation that occurred, or reinnervation, or maybe more than likely kind of avoiding a loss of the denervation or slowing it. So we quantified some of this by looking at axon perimeter and neuronal perimeter and neuronal area using a program called NMJ Morph. And we can see that the drug didn't show a statistical difference from wild type, but it didn't really improve it from the untreated animals. So it was a little muddy. We put together a lot of the variables that we measured for neuromuscular junction, along with some of the physiologic variables, did a PCA analysis because that's independent of the actual measure that you're using. And we can see here that the red dots are the untreated CX32 def, the green dots are wild type, and the blue dots are the drug-treated animals, suggesting that they are clustering with the wild type. So our conclusions are There's a good correlation between this nerve conduction improvement and grip strength in most of the models. This may be related to the slowing at the neuromuscular junctions, but we don't know yet. Provides first proof of principle that sildenafil therapy may work in gain of function CX32 mutations that cause trafficking defects. And the gradual loss of efficacy upon washout may be more related to probably indirect anti-inflammatory effects rather than the direct changes in nerve morphology, but we need to look at this a little bit more. As I said, the lack of change in neurofilament light chain was a little bit disappointing. I didn't show you the data where we made HSP70 knockout mice on the CX32 def background. If you treat those mice with, I'm almost done, with sildenafil, they do not respond to the drug, demonstrating the requirement for HSP70 for drug efficacy. So the clinical perspectives for this disease modification strategy is first in class for this use of targeting molecular chaperones to treat neuromuscular deficits in CMT1X. Drug has rapid translational potential since GMP grade drug is already available because a phase 2 trial is currently recruiting and in progress through our Reata partnership. And this may provide a pharmacologic alternative or supplement to patients awaiting or ineligible for or poor access to promising gene therapy approaches. And I'm done. Thank you.
Time for 1-2 questions before we move on to the next speaker.
Always good to wow your audience. Yeah.
Yeah, I can ask a question actually. I was wondering if you might touch a little bit on your preclinical data comparing the diabetic neuropathy results to the CMT results. Yeah.
Well, we haven't run them together. I haven't made any CMT mouse diabetic and taken a look at it. But from a functional standpoint, the efficacy data is very similar. And when we make HSP70 knockout animals diabetic, treat them with the drug, it doesn't work as well either, or it doesn't work at all. And best comparison is the nerve conduction velocity, which shows similar improvements, almost back to baseline in both models.
Any questions? Awesome.
Thank you.
Oh, wait, Dennis?
Thank you. Because you mentioned in that you think it might be anti-inflammatory effects. Did you look at anything like macrophages or something in the nerve? Or
I'll show you the slide later. It's in there, but I knew I wouldn't have time. But yes, it is. We did change the M2-M1 ratio.
Small question. And was the CMT
Was the CMT or the connexin 32 knockout that had a more severe phenotype than the point mutations? Or do you recall?
You know, Charles might be the better one to answer that. I mean, I think just based on how the animals are, they all are pretty similar.
Yeah, because they're quite mild phenotypes.
Right.
I think,
Yeah. And you could look from the grip strength that the values were fairly similar between all the mice when they kind of started the study at 4 months. And so I don't think that when we initiated therapy, the animals were that different. Now I think there's probably differences in the progression of the disease. I think that the knockouts progress quicker and worse than the gain of function mice.
Then,
but you know,
could
it's still protected. Yeah.
Could you speculate on why the therapy is more effective than in the knockout versus the point mutation? Because you would assume it would be more effective in the point mutation. I think so anyway.
Well, that was our hypothesis.
Oh, yeah.
That it might be less effective because of the formation of the protein aggregates. And now HSP70 has multiple jobs. One, helping to clear protein aggregates. The other, to help promote what it's doing to promote the neuroprotection. And so we were fortunate that we still saw efficacy. Now, the level of efficacy may be improved by increasing drug dose it can increase frequency, I guess, because we're only doing once a day, but we haven't gone any higher than 3 mg per kg. And from a human perspective, the 1 mg per kg dose is right about where you would dose a human, which gives us a very good response.
Thank you so much, Dr. Dobrowsky.
This one works just, oh, there we go. Thank you. I'm Albert Misko. Thank you very much for the opportunity to talk. I really appreciate it. I finished my PhD under the mentorship of Bob Balow at WashU. When you're looking at over here, 2 axons that have been teased out of a nerve fiber, and we stained them. Little green dots are mitochondria, and the blue outline are the axons. And this is what we were really interested in when I was there studying. How did mitochondrial dysfunction really relate to axonal integrity and degeneration in CMT? Now, full disclosure, going forward, I went on to train as a pediatric neurologist. Everything I've done since then, gene therapy and gene editing for neurodevelopmental disease. But I hope that my background at least qualifies me to give this talk. I'm really happy to be here. Also, right now, currently, I am at Novartis, but I'm not here on their behalf. This is talking completely outside of my work. I'm not involved in the DTX project, and I can't answer any questions. Okay. Great. So why mitochondria? Saying that mitochondrial dysfunction is involved in the degeneration of axons is correct, and probably analogous to saying that the degeneration of neurons is involved in neurological disease, right? They're very broad. And so there's a lot of topics to talk about and very hard to cover, which is why I bring it up. Mitochondria are important for generating ATP through the electron transport chain. They have a number of anapleretic reactions that feed metabolites into the Krebs cycle to keep a lot of different processes, including recycling of neurotransmitters going. They buffer calcium. They're the spot where iron sulfur clusters are made, those cofactors for other enzymes. So there's a number of different functions that these mitochondria are playing. And as you can imagine, they're very important in a lot of different tissues. In fact, in some of the most profound mitochondrial diseases, they're not axonopathies, they're not neuropathies, right? They're diseases of the brain, like in Lee's disease, or of the muscle, these very energetically hungry tissues. But there are cases in which genes that are involved in mitochondrial function directly lead to very prominent length-dependent neuropathies. And the question becomes, why? Is it, one, it could be that some of these genes are maybe only expressed in peripheral neurons and they're not compensated, or there's not redundancy. Another thing could be that these are very interesting in terms of their structure cells, right? Like if you had a nerve cell body the size of this room, the axon would probably go from here to Baltimore. So the actual mass of the cell in the axon is huge in proportion. And there are probably very unique functions of mitochondria that relate to that special morphology or structure. When we were looking at the types of different genetic disorders or gene mutations, gene variants that lead to CMT, looking at the mitochondrial ones, MFN2 is high on the top, along with GDAP1. These are known to be involved in mitochondrial fusion and fission. And then there are other genes that are involved in metabolic pathways in the electron transport chain. We decided to focus on MFN2, and I think for this talk, I think dynamics is an interesting aspect that really relates to a propensity for axonal degeneration in CMT. So thinking about mitochondrial dynamics, how do they work? Well, for the most part, we know a lot of mitochondrial biogenesis happens in the cell body. Potentially it happens elsewhere, but I think that's still controversial. And so these mitochondria have to move from the cell body all the way down these axons. So not in me, but in someone taller, that's a meter, right? Like all the way down from your dorsal root ganglion to the bottom of your foot. And that's a long way to go, especially at the speeds these things travel. So it's very important that you're able to transport the mitochondria. At the same time, they undergo fusion and fission. This is really important because they're able to swap out nucleoids or the DNA material that may have genetic mutations, or if they've accumulated mutations, also oxidated proteins, other stuff that's gone bad that can then undergo autophagy or mitophagy to get rid of it. So it's also like a quality control process. And if you're not undergoing fusion and fission, your mitochondrial function is compromised. So those are the main, things that we were thinking about, and we were wondering, how does MFN fit into all of this? And well, we'll jump ahead, fine. So at the time, it was when mitofusin was very important for undergoing the fusion of mitochondria, especially on the outer mitochondrial membrane, that first linkage. In the course of our studies too, we actually figured out that it was involved in directing transport as well, as it interacted with some of the transport complex and interrupting it. There seemed to be a very intricate relationship between transport and fusion and fission. So thinking also a bit about the disease, right? This is CMT2A is a length-dependent neuropathy. So you're really getting involvement of the longest axons first, and they're the ones that are the most susceptible. So why are all these things playing a role in the longest? How are they most susceptible, or how are the longest axons susceptible to dysfunction in the fusion or perhaps the transport? So when Bob first took a look at these are from mitofusin 2 wild type expressing DRG neurons, or DRG neurons overexpressing a dominant negative form of MFN2. There was an abnormal distribution of the mitochondria in the soma and along the axon. And if you look over at the time-lapse microscopy down here, in the wild type, you'll see the mitochondria moving back and forth. There are two types of movement. There are fast movements and there are slower moving mitochondria, or there's two different populations. But when you come down to the, I guess I'm not going to be able to do that, am I? Yeah. I think it for, oh, there we go. Is there any chance you could click on the lower video that says R94Q? Well, maybe you can take my word for it. They don't move. They're actually just kind of like bobbling around a bit, and they're not distributing properly throughout the axon. There you go. Oh yeah, there we go. Thanks. So there's just this little guy bobbing around, right? But for the most part, you have these segments of axons that are not seeing any volume or mass of mitochondria pass by them. You can look at that in something called a zymography too, or this, zymograph, sorry, where you're looking at the movement of mitochondria. These are anterior grade movements, these are retrograde movements, and the ones that are standing virtually still. And again, with R94Q, this dominant negative, you're not getting the appropriate movement. And there's a sort of a shift in the population of the size distribution of these mitochondria because they're smaller. So they're not moving and they're not undergoing fusion or fission really well. I don't know if you can see this very well. It's more clear on the screen there, but if you overexpress some of these mutants that cause CMT2A2, you see these like kind of big blobs there. Well, I'm going to show you in a second what those are. Oh man. Okay, so big blobs. We saw these dilated axons that weren't present when you had the normal MFN2 expressing. So if you do time-lapse microscopy on those and you look at calcium over time, and let's see if this works. There we go. Okay. And then this one too here. Basically, over the course of 24 hours, what we found is that we could find segments of axons where there were no mitochondria. Calcium increased at some point. There was a bleb in the mitochondria, or bleb in the axon, I'm sorry, and then they pulled apart or degenerated at that time point. And that blebbing is something that you can see on histology too on a lot of different types of axonal neuropathies. So we knew that we had a good model at least about CMT2A, MFN2 and mitochondrial transport and fusion were important, and we could model the dynamics, but what do we do about it and how do we think this is working? Well, at this point, this is what we were assuming. In a normal axon, mitochondria are moving around. And at some point, there's some kind of insult. Maybe it's increased calcium, maybe it's the need for ATP, or whatever else the mitochondria is doing. And if it's not able to do that, or if it is able to do that, then the calcium is buffered and it's fine. But in a sick axon, where the mitochondria aren't moving properly, if you have that insult arise, it's not going to be taken care of because there are no mitochondria that are able to get there, or ones that aren't viable enough to deal with the insult. And at that point, it gets out of control. It's basically a fatal defect, and you get degeneration of the axon. These are stochastic events most likely, and that could explain the length dependency too. In addition to the idea that there's long transport, it could also just be that the longest axons have the highest chance of incurring some kind of injury along the longer lengths. All right, and now what to do about this? Well, one thing that was kind of interesting that we saw while we're doing these studies was that we could increase MFN1, which is another isoform of mitofusins, and rescue the nerves in vitro. So in vivo, Bob's lab then went ahead and made this MFN2 overexpressing mouse to model, or in vivo to model the disease, and indeed decrease survival. And what you're looking at is neurological dysfunction here. So latency to fall, grip strength, both worse in this mouse. And then the mouse, you're also seeing decreased visual acuity. These are not C6 N strains, which all have the RDB mutation that they all go blind. This is optic atrophy due to mitofusin mutation. And in these mice, you definitely get axon degeneration without loss of cell body. So good model of an axonopathy. Mitochondrial clustering and loss of transportation or loss of mobility also occurs. So it mimics what we see in vitro. But like I told you, we could take MF1 and overexpress it. And when we do so, we actually rescue, these are all a distribution of the speeds that they're moving anterogradely and retrogradely. We're able to rescue mitochondrial mobility and axon viability in vitro. Now that was cool, but it turns out that you can also do it in vivo. So crossing the MFN2 dominant negative expressing mouse with an MFN1 overexpressor also was able in vivo to reverse the neurological phenotype of that mouse. This became cool because it gave us a new venue to go after. One, could you deliver more MFN1? Or two, could you upregulate MFN1 because it's not really expressed in a lot of the nervous system. So indeed, it rescues the mitochondrial phenotype, the neurodegeneration, and there's something else I want to highlight here. So what this is a balance, right? We're trying to augment transport or fusion of these mitochondria. Can we do this in a different way? I want to highlight some of the awesome work by Gerald Doran's lab, where this mitofusin 2, the way it acts is there's two conformations. In the passive state, it acts as a tether, like I said before. And then in the active phase, this GT-PACE, it allows a conformational change that facilitates mitochondrial fusion. And Gerald Doran's lab had come up with some small molecule allosteric activators of MFN2. And this is really neat because remember, it's a dominant negative disease. So there is one good copy around. And if you can augment the ability for it to facilitate fusion, maybe you could save the disorder. And that's what they were showing. And that's what they found in their own rodent model of CMT2A. With one of the MFN2 activators, they were able to restore a lot of the mitochondrial mobility. And there was functional recovery in the mouse too, or prevention of disease in the mouse. So this is a really cool idea that perhaps, we can increase the function of mitofusin to protect these mice. So I want to leave you with some of the real questions that I think that are important in understanding this. If you think about all the forms of CMT, mitochondria are going to play and dysfunction are going to play a role. But is it a primary driver of disease pathology, or is it a downstream secondary effect? If it's a primary driver, addressing it probably is going to have a big impact. If it's a secondary driver, it could ameliorate disease for sure, but probably to different extents and different pathologies, depending on how much of a role it's playing. There are other pathways and other things that are going on. A good example, just take for an analogy, NMDA receptor excitotoxicity, which we know a lot about in neurodegenerative diseases. Mitochondria always play a role there too. If you poison the mitochondria, you get NMDA-related excitotoxicity, too much glutamate export, and then death of the cell. At the same time, if you poison the cell in a different way and it's not directly mitochondrial, say you put in a lot of glutamate on the cells, you also get mitochondrial dysfunction because it's part of this feed-forward loop. And I think it's really cool because Jeff's work was really highlighting how there's another loop that's very similar in the axon, though there are different drivers for it. And again, the question will be, in these different forms, how much can augmenting or affecting mitochondrial dynamics really prevent the disease? Also, are there other forms of mitochondrial dysfunction that are affecting this besides the dynamics? And another fascinating thing I think is, when we were expressing MFN2 and MFN1 in these cells, this is anecdotal, but man, they look healthy. It almost looked like they were protected. You could throw on a lot more toxin onto those cells. And in this case, it would be vincristine. We were using ionophores, a bunch of different things to poison the cells, and they were more resilient. So I think another question is, can you actually soup up the mitochondrial activity in these nerves to sort of have more reserve capacity and more resilience to a disease process that's going on? And that's it. Thank you very much.
Yeah, very nice work. But when you saw the calcium influx and you were closing it like a blebbing in the axon, we see very similar things. We call them varicosities. Okay. We basically think that they're misfolded proteins that are aggregating in the axons that are inhibiting the transport, okay, down. Do you know it's a calcium influx? Do you think it's something different, or it's just your thoughts on it and you really haven't characterized it directly, and it possibly could be varicosities?
Absolutely, great question. Calcium influx could be corollary, and it's definitely a phenomenon that we saw. Where else are varicosities seen? One thing that jumps to mind I thought was very interesting are stretch injuries. So back in I think the 50s to 70s, there were good studies where they took cat nerves, right? And they would stretch them and put weights on them. And indeed, you could actually induce these varicosities as well. And they were due to the contraction of actin. And then that actin contraction, whatever, it triggered downstream from there, including calcium influx, I believe, led to axonal degeneration and injury. So I think it's like a loop, some kind of feed-forward loop. There's a program that's going on there. Definitely, maybe misfolded proteins have a role in that as well. And
Because we see them in the disease model.
Okay.
Compared to the wild type to the disease, you see a lot more varicosities in the ALS models and some of the other diseases, the CMT models we're looking at, as compared to the wild type. And we think transport, okay, from the cell body to the synapse is one of the issues that's being compromised in these disease models because of these varicosities.
Oh, interesting. Oh, that's really interesting. Yeah. Thank you.
Related to that comment about the varicosities. So I think of mitofusin as regulating fission and fusion. So how is it disrupting transport so bad when it's not there?
Great question.
Or mutated?
So one of the things that we found is if we took about 7 to 10 different mutants of MFN2 that had been identified in patients with CMT2A. We knew from David Chen's work at Caltech that some of those mutations disrupted mitochondrial fusion while others did not. But when we overexpressed them in the DRGs, they all disrupted transport. So we don't know exactly how that was working. The closest we got to an explanation was showing that mitofusin 2 binds to MIRO. MIRO is a calcium sensitive regulator of mitochondrial dynamics and transport and its attachment to kinesins. So perhaps it plays a direct role in regulating transport as well as fusion or fission, or perhaps transport and fusion and fission itself are more intimately related in a way, I don't completely understand.
It's very interesting what you just said. I was wondering, you have an amount of mitofusin per mitochondria, but mitofusin can tether several other proteins in several other compartments. Do you believe, or do you have reasons to believe that the mutant would get stuck on a given interaction? Like the endoplasmatic reticulum is the main organelle in the axon, preventing from tethering mirror or other proteins. Have you seen that? Have you come across something on those lines?
Not to my knowledge, no. I'm sorry, it's been a while too, I gotta admit. It's been a good 15 years or so. But.
I have one question. This is from an online member. Do you have any experience with heterozygous FBXL4 plus heterozygous SDHB causing mitochondrial dysfunction in CMT2?
Gosh, I'm sorry, I don't know.
Yeah, that's going to be difficult.
I apologize.
Okay.
All right. Well, thank you so much, Dr. Misko. Lastly, we have Dr. Dennis Klein.
Okay, thank you, and also for inviting me and giving me the opportunity to talk today about this in this convention about inflammation as a common disease mechanism in CMT diseases. And I directly want to start with the concept of the group and the basic hypothesis. And this is that the reason for a quite similar disease outcome of primary distinct mutations might funnel down in common pathomechanism. Should I? Okay. Okay, might funnel down in a common disease mechanism. And we think that one of these common pathomechanisms might be inflammatory reactions, as you can see here highlighted in this representative image, is showing an increased activation of macrophages in CMT1 mutant nerves. And if you can indeed show in these models that inflammatory reactions are present and functionally relevant, and you can also see similar observation in patient biopsies, so also increased macrophage activation, you might of course have now the opportunity for a therapeutic intervention, so modulating these inflammatory reactions and thereby modulating, so mitigating disease outcome. And we could indeed show in several studies here shown, or starting with genetic proof of principle crossbreeding approaches, that neuroinflammation, so here really talking about only about the macrophages in the CMT disease progression, are a cause and not a consequence. And this was shown by crossbreeding approaches using the P0 heterozygously deficient mice as a model for CMT1B, so the CMT1 demyelinating type, and those were crossbred with this osteopetrotic mice. So these osteopetrotic mice, they lack a specific cytokine that is important for macrophage activation. So you basically block macrophage activation in the absence of this macrophage colony stimulating factor, as the old name was. And this is exactly what you also observe when you look in the peripheral nerves of these mutant mice, and that this typical increase in the number of macrophages that you can quantify here using FR80 as a marker. Previously, we have heard CD68 as another macrophage marker that can be used, and you can see that with the lack or upon the lack of CSF1 deficiency, you prevent this increase in the number of macrophages. So how about the disease outcome when you target this genetically? And here you can see on these ultra-thin sections in these P0 heterozygous deficient mice that this genetic lack of CSF1 clearly substantially ameliorated the neuropathy. And just again, as a reminder, so both have the CMT1B mutation, just these mice or the nerves of these mice, they lack this macrophage activating molecule. And as we could also show this, this is not only due to the CMT1B model, but also in connexin deficient mice as a model for CMT1X, as we have also heard. And recently also in the most common form, so the CMT1A as a PMP22 overexpressing mouse model, showing this impact of the macrophages. And so our group could identify over the last years really molecular and cellular interaction in the diseased nerves of the CMT1 mouse models, highlighting this role of the CSF1 activated macrophages. However, of course, based on these genetic approaches, there were questions emerging regarding a more translational approach. So is there any treatment strategy possible? And therefore, we used also quite a similar approach, not this CSF1 receptor antibody, as Jeffrey Milbrandt has shown. We used the compound PLX5622, which is also CSF1 receptor inhibitor designed by the company Plexicon. And basically, if you apply this drug to these mice, you would target or block the downstream signaling of CSF1 receptor expressing cells, among these also, of course, the peripheral nerve macrophages. And what we did in this treatment approach, in this long-term treatment approach, over 9 months of treatment in the distinct CMT1 mouse models, so connexin, P0, and PMP. So all these 3 demyelinating forms was a treatment with this PLX5622, and you can see in the representative images here on the left, but also in the corresponding quantification that shown here for this connexin model, but this is also true for P0 and PMP mice, that you reduce drastically the number of these nerve macrophages. Here also the question, okay, targeting macrophages, you can reduce the number of macrophages. How does this translate to the disease outcome, nerve phenotype? And there we were also able to show that they clearly improved the disease outcome. So on the one side, on the left, you see the nerve structure. Here, the untreated controls at 12 months of age compared to the PLX-treated mice femoral quadriceps nerve, as we have now several times heard at 12 months of age, and also based on the neurographic recordings that you can see on the right side, treated mice showed an improvement compared to the untreated controls. In several other studies, we focused on other relevant questions for translation, and these focused on one side on the time point of the treatment, because we were able, as we previously failed to show any impact in beneficial effect upon macrophage targeting in this C61 model, we could demonstrate that there is indeed an effect to mitigate the neuropathy and also ameliorate the nerve function. However, there is an early treatment necessary. So there is a critical time window where you need to treat these mice with a PLX, so with the CSF1 receptor inhibitor. And in another study, we could also demonstrate that there are similar therapeutic time windows in other models. However, and this is even a more important aspect of this study, was that we investigated kind of the sustainability of the treatment. So we observed a preserved treatment effect. So when we treated these mice until 6 months of age, then we terminate or stop this PLX treatment and investigated these mice 6 months later. Here you can see shown by the representative grip strength approach that here these mice on the right side that were with the terminated treatment, they were at the same showed the same preserved grip strength compared to their wild type controls, and those that received the treatment early onset and throughout the whole life compared to untreated controls and those that received the treatment at a later time point. And these observations were also visible in the CMT1X mice. So we concluded that this macrophage targeting might be a future translational option, however, keeping in mind this early treatment, but with sustained beneficial effects. So it doesn't mean that you would need to treat, as we have heard before, early and continuously, but early or like as this preserved treatment effect would be possible, you don't need to treat the patient throughout the whole life. These studies focus more like on this demyelinating CMT1 forms. So of course the question emerged also, how about more axonal CMT forms? And this is a question that we try to resolve in the CMT-RF funded project. And therefore we used a model for the CMT2J, which is due to a substitution in the myelin gene protein 0. So also mutations in a myelin gene that do not automatically result in a demyelinating CMT1 form, but can also result in an axonal form, as it was described in these patients. And we used a mouse model that was recently generated by the group around Larry Roberts, Laura Feldry, and Maurizio Di Antonio. And indeed, when you look at these mice and you use the hemizygous mice, so carrying only one allele with this mutation, similar as it was described in the patients here on the right side, you can see that these nerves, peripheral nerves, look rather normal at 12 months of age, but at 18 months of age, interestingly, they really develop this late onset axonopathy. So you see degenerating axons, you see fibers with periaxonal vacuoles or condensed cytoplasmic structures. So all pointing to axonal perturbation. And interestingly, you can also see the presence, again, of these activated macrophages. So we asked the question whether modulating the macrophages or depleting the macrophages with this PLX compound, PLX 5622, would again be a suitable therapy to modulate inflammation. So if macrophages might also play a similar role in these axonal CMT forms. And here you see the treatment strategy. So we treated these mice starting at 12 months of age, where there's, as I mentioned, this relatively normal peripheral nerves detectable until 18 months of age, where you see the signs of axon degeneration, similar as it has been shown in the patient biopsies, as you can see it here on the right side. And then at 18 months of age, these mice were analyzed. Of course, we first focused on the target cells, in this case, the macrophages, and we could see, similar to our other treatment studies, shown by the representative images, but also by the corresponding quantification, you kind of prevent this increase in the number of endonuclear macrophages here significantly. And the same was true when we looked at these activated phagocytosing macrophages, where you can also see that they were basically not present in these peripheral nerves, comparable to their wild-type littermates. So how about here the disease outcome? And there we were really surprised, to be honest, because as you can see by the semi-thin sections on top and ultra-thin sections below, basically by targeting the macrophages, this disease doesn't develop, or this typical histopathological alterations that you can detect in the nerve at 18 months of age, they are just not present. And this was at least in our hands with this PLX study, so the most drastic effect after a CSF1 receptor inhibitor treatment. And of course, you can quantify these features, these histopathological features, and can see that those like signs of axonal damage, so degenerating fibers and so on, also regenerating clusters, they are really at the level of their wild-type littermates of 18 months of age. So really demonstrating this stop of disease progression or prevention of disease development after the CSF1 receptor inhibitor treatment. And the same is also true when you look at more distal aspects. So also abnormally innervated neuromuscular junctions were significantly reduced after the macrophage targeting. As a functional readout, we looked at these beam walk studies where the mice have to cross this narrow beam and you measure the latency, but also the number of foot slips. And here, at least for the number of foot slips, you could also see that this increased number is significantly reduced upon macrophage targeting. So as a take-home message, and also thinking about future directions, we conclude that the macrophage targeting is really a future translational option, but not only for distinct CMT1 models, but also for this CMT2J model. However, as mentioned, there are still some urgent questions regarding this P0 TM mouse model, especially focusing also on the time window. So is there also a critical time window regarding treatment onset and the duration? Also about the mechanism. So how does macrophage targeting prevent this initial axon degeneration? Maybe a question similar to the psammopathy, what we have seen before. And another point, and this is highly important, I think also for the future direction regarding also drug discovery or drug design studies is the molecular characterization of these nerve macrophages. So what are really the molecular features of these pathogenic macrophages that promote the disease? And to do this, we use currently in another project the techniques of the single-cell RNA sequencing technology, so this 10x genomics that some of you might have heard of, or many of you might have heard of right now. And we used in this study, or comparing the peripheral nerve macrophages of wild type versus this CMT1B mouse nerve macrophages, so from the model of the P0 heterozygous mice. Here you see the schematic workflow and the analysis. And we are currently analyzing this data, but already saw that there are different dynamics, different types and clusters of macrophages appearing or disappearing between wild type and mutant. So there might be a more homeostatic cluster, more disease promoting cluster, here highlighted in red versus green. And you can now, of course, look at these target molecules. You have to validate those. And I just exemplarily took the CD163, which is a scavenger receptor and higher expressed in this rather homeostatic cluster in the wild type. And you can transfer this, of course, also to other models. So not the CMT1B, but now transferring this to the CMT2J model. And actually, this is what we did. And you can see in this representative images that this number of CD163, so double positive macrophages here highlighted in yellow, so these rather homeostatic macrophages are shown in a higher frequency in this femoral quadriceps nerve compared to this TM mutants that I showed you before. However, there are still like further validation steps in progress. And with this, I want to thank the group, especially to Rudolf Martini, who's also attending online, collaboration partners, the sponsors, and you for your attention. Thank you.
We have some time for questions. Okay, it's working now.
You. Hi, Dennis. Great talk. I'm Kelly Lingard. I'm at Loyola Chicago, and we've been really interested in your work and interested in the idea that these immune cells might serve as a drug delivery targeting mechanism. And I'm wondering with macrophage inhibition, if there's any risk of general immunosuppression or like a risk of infections.
Yeah, so that is a good question. And I think one of the most, or biggest hurdles to overcome for translating this. At least in our treatment studies, we didn't observe any side effects. These mice were monitored and so on. But of course, you need to be aware. And this is also shown, of course, when you use this kind of inhibition, you would target not only the nerve macrophages, but also other CSF1 receptor expressing cells, like the microglia, for example, in the CNS that have also many important functions, homeostatic function, synaptic pruning, and so on. So these studies serve also kind of proof of principle treatment strategies. But I think that is then the goal also for future drug development to and the question to get really specifically into these peripheral nerves and maybe specifically target these disease promoting macrophages, because not all of these macrophages, we have seen it maybe also in this last immunohistochemical staining, there were still like homeostatic macrophages if you would use this CD163, for example, as such a marker in the nerve that might be targeted also, of course, with this kind of treatment. So I think that is the really the basic question, how to have the basic issue to get really a specific high specificity, especially if one would need to go to a rather early onset treatment, then you need to have something safe. Yeah.
All right.
Question over here, Edritz. Would you like a mic? Oh, sure.
Hey, Dennis, that was a really lovely talk. I was wondering if anyone has ever sort of formally looked clinically at patients with CMT 1A, 1B, 1X, and 2J with seeing sort of how their CMT responds after exposure, sort of incidental exposure to any immunomodulatory treatments. Like, has anyone ever looked like when CMT1A patients get steroids for some other condition, if that affects their CMT at all?
Yeah, that's a good question. Maybe I can show you maybe later also some slides on this, but in principle, they're like normal, I would say normal CMT1 type in the clinic systematically. I think there are not really a lot of studies well performed or ongoing. When there was some immunomodulatory treatment, this was most often the case for CMT patients that had something on top, like a CIDP or something on top. And I think then there were also immunomodulatory treatments that showed effects in some cases. But also for this normal, I would say normal CMT1 types, there are indeed, but just rare cases, also biopsies from nerve biopsies from children where you see indeed that similar activation of macrophages. So macrophages phagocytosing the myelin. So similar things but also at the rather younger age in the patients. Yeah. But there is, I think, not really a lot of things going on, and I think that is also something that has to be changed or should be more investigated in the future.
Rick, I'll pass on the last question. Just one last question and then we'll move on.
So Dennis, if you looked at microglial populations and CSF1 receptors important for those cells?
Yeah, so when you use this PLX compound, you deplete also the microglial cells to a high percentage, and it's used for many CNS studies, but has also then of course this effect in the CMT model, and that what I mentioned is really serves as a proof of principle approach for really showing also pharma industry that this pathway would work and would be worthwhile to focus on, but we really need to have a high specificity, I think.